Well, cat breeders are aware that kittens expressing the X-linked calico coat pattern and tortoiseshell pattern are almost invariably females. Why are they certain of this? Because they breed lots of cats and they're always females. So they don't even need to know much about genetics if they're breeding cats, right? They almost never see a male tortoiseshell or calico cat. So, but a geneticist can be certain of it, right? So we should be certain of it in our class, and we don't even need to be cat breeders. And so let's take a look at why females are almost always, cats with calico cats and tortoiseshell cats are almost always females. So in class we talked about, and this is problem 7.22. So in class we talked about how, cat coat patterns are really interesting, and for the most part we only need to know about one gene. We're going to call this gene C, and this gene C has two major alleles, one which confers the red pigment, which is kind of that orange color. I don't know, breeders call it red. Kind of looks kind of orangish to me. So a red color, and allele CB, which is for a black color. So a red pigment or a black pigment. And then you have gray and things like that, and tan, and those are all due to other genes which sort of dilute these pigments and lead to different shades of black or red. Okay, so but we're only concerned with the red pigment and the black pigment. Okay, so these happen to be X-linked. This is why we're talking about it in this chapter. So these are X-linked. And as a result, if you have, let's say our male genotypes can be CR with a Y chromosome, CB with a Y chromosome, and that's it. And these males here are always going to be sort of that red-orange color, or maybe they have the orange base. But okay, so red or orange, and these are always going to have black. These are always going to be black, so they're never going to be orange and black, like with a calico or tortoiseshell. So females, however, can be CR, CB, CB, or CR and CB. Now these are always going to be red. There's no choice there, right? There's no black allele. These are all going to be all black. So all red up here, all black down here. And these are the ones we're interested in. So notice how these are heterozygous, and males can't be heterozygous for X-linked genes. They are hemizygous. So these females right here have both alleles, one to produce red, one to produce black. And it's hard for us to know which is dominant over the other one because of this thing called X inactivation that we just talked about in some of the earlier problems. So X in inactivation. So in each cell, one of these is going to be inactivated. So during the first 1 to 3 weeks, and maybe that time might be a little different for cats, neither X is going to be inactivated, but then we're going to end up with lots of cells, thousands of cells, with both of those activated, and then one of these chromosomes, X chromosomes, is going to be inactivated. And let's say in this one and this one, the X chromosome with the black allele is inactivated, and it's put into the bar body. And the red allele is still active because it's on the active X. And in these down here, we'll say the X chromosome carrying the red allele is inactivated. And the one carrying the B allele is still active. Okay, so every time these cells divide from now on, the black allele, this one right here, the X chromosome with the black allele will always be inactivated. Every time these cells, every time a cell with a black chromosome inactivated, with the X chromosome carrying the black allele is inactivated, the daughter cells will also have the black chromosome, the X chromosome carrying the black allele inactivated. Okay, so same thing for cells which have the X chromosome carrying the red allele inactivated. So I've diagrammed the nuclei here. So let's say that undergoes mitosis, and we've now got 2 cells with nuclei here, and each time the X chromosome with the red is inactivated. So once an X chromosome is inactivated, it stays inactive in all of the daughter cells following each mitosis. So if you have a cat here, here's our cat, and if you've been coming to class, you know I'm getting a little bit better at drawing these little animals. Here's a tail. Okay, here's our cat, and if we have a patch here with a bunch of cells, all these cells are probably, the closer the cells are together, the more likely they came from the same parent cell. So that's why we can have a patch of cells here, pigment cells, that all produce, let's say, black. So we got a black patch there. Now let's say there's a patch over here. We have a bunch of cells that all started from a cell that had the red allele, no, the black allele inactivated. So all these would be red. So, and we could do the whole cat, we could do the whole thing to the whole cat that way. And this would be a calico cat where the patches are nice and big, and the reason the patches are nice and big is there's another gene which kind of slows the rate of migration of the cells and keeps them from far apart from one another. So this is why we don't have small patches of red and black in the calico. But in the tortoiseshell, well, they don't have that particular, that other gene that controls that slows the rate of migration. So you can have little patches of black next to little patches of red next to little patches of black, and that's a tortoiseshell cat. And you should look up some pictures of tortoiseshell versus calico to see what I'm talking about. But the colors, the patches are closer together in the tortoiseshell. But the concept is the same. We have X chromosome inactivation leading to the patches of red and black. And why can't we see tortoiseshell or calico in a male? Well, it's because males only have one X chromosome. So that same X chromosome is going to be active in all cells of the male. So let's go back up to the male genotypes. No X chromosomes are inactivated in the males, so they can only be all red or orange or all black. Now there is the rare case of the Klinefelter cats, right? Klinefelter, which are X, X, Y. Now in this case, you could have an X chromosome that has the CR allele, and another one that has the CB allele. And these cats right here would be either tortoiseshell or calico. However, they would be sterile because Klinefelter, the syndrome, is associated with sterility too. So you can occasionally get a Klinefelter cat, and so once in a while you will see a male calico or tortoiseshell. And I sent a story out about how a vet, I think it was 54-year career, just recently saw his second male calico. So it's really interesting. Okay, so this is that's the answer to 7.22.